Hello everyone, welcome to this video. On this video, we will tackle the different disaccharides, the bonding or the linkages that connect those disaccharides, and we will also take a look at the different polysaccharides, their occurrence, their properties, and we will also take a look at some carbohydrates derivatives. So the first thing that we will see is the disaccharides. Disaccharides are carbohydrates in which two monosaccharides are bonded together by a glycosidic bond. So take note that a bond that connects the two sugar units is called glycosidic bond or glycosidic linkages. So as you can see in this figure, there are two monosaccharides. Let's label this as one. Then this is another glycosidic. This is another molecule of monosaccharide. At the moment they are separated. However, when the two are joined, they become a disaccharide. So you notice the formation of glycosidic bond here. And in the process, water is removed because as you can see here, one hydrogen, the hydrogen here is removed, and then the hydroxyl group here is removed. That's why that is equivalent to water. So the first example is maltose. It says it is often called malt sugar because this is intermediate product of starch degradation. Starch is a polysaccharide. It's made up of many glucose molecules. In starch, the glucose molecules are linked together by glycosidic bond. And in starch, the glycosidic bond is alpha 1,4. So maltose is also joined by the glucose molecules in maltose are joined by alpha 1,4 glycosidic linkages. So let's take a look how or what is an alpha 1,4 glycosidic linkage. So here in the molecule or maltose is composed of two glucose molecules. So here you have alpha D-glucose and alpha D-glucose. Remember that when we say alpha D-glucose, the OH in carbon number 1 is written or shown downward. Okay? Likewise here, the hydroxyl group in carbon number 1 is downward. Okay, so remember when we say alpha, that's down. With this beta, okay, the bond or the OH points upward. Alright, so let's connect the two glucose molecules. So it says here the specific linkage is alpha 1,4. So the bond in carbon number 1 is written downward. Alright, that's why alpha 1. So the bond in carbon number 1 is downward. And then remember here, the hydroxyl group, which is highlighted, is removed, and then the OH, the hydrogen here is also removed. So you make a bond from carbon number 1 downward, and then just connect it with the following molecule as is, because the hydroxyl group here is pointing downward. also. However, the alpha and beta only applies to carbon number 1. Okay? So, the maltose is hydrolyzed by an enzyme maltase. So, this maltose, what happens here is it can still be hydrolyzed by an enzyme maltase. So maltase breaks the glycosidic bond. Okay? So what happens when this glycosidic bond is broken? The products are 2 glucose molecules. Okay? So that's what it means by hydrolysis. The bond is cleaved or hydrolyzed by enzyme maltase in the presence of water. Another disaccharide is cellubiose. Cellubiose is an intermediate product in the hydrolysis of cellulose. Cellulose is a polysaccharide, and just like starch, is made up of many glucose molecules. But this time, the glucose molecules in cellulose are linked by beta 1,4. So, as you can see here, although it's made up of 2 glucose molecules, the bonding is different. So here, as you can see, the carbon number 1, the bond in carbon number 1 is upward. As I said earlier, when we say beta, the bond is pointing upward. Okay? And it's connected to carbon number 4 of the following molecule. So as I said earlier, the alpha and beta only applies to carbon number 1. So what happened here is we remove, in the previous example, we remove the OH or the hydroxyl group from this carbon, and then we remove hydrogen from the OH here, and then this oxygen serves as a bridge between the two monosaccharides. So this link here is what we call beta 1,4 glycosidic bond. Lactose is another disaccharide, but this time this is made up of beta D-galactose and D-glucose. So here is the structure of beta galactose. As you can see, beta is up on carbon number 1 connected to carbon number 4. So the first molecule that has to be written is the galactose, okay, and then followed by the glucose. So the connection is beta 1,4 glycosidic bond. So lactose is the major sugar found in milk, and once lactose is taken, or when we drink food containing lactose in the small intestine, this bond, the beta 1,4 glycosidic bond, will be hydrolyzed or will be broken down by enzyme lactase. This lactase is present in the small intestine. So when this bond is broken, the beta 1,4 is broken, what will be produced is equal amount of galactose and glucose. So remember, the specific bond in lactose, it's beta 1,4 glycosidic linkage. This time, we have sucrose, or it's commonly known as table sugar, and it's composed of alpha D-glucose and beta D-fructose. So this time, as we can see, the glucose is bonded to fructose using its carbon number 1, and the carbon number 1 is pointing downward. That's why it's called alpha. And the fructose uses its carbon number 2. Now, the link here is unique because the two sugars, the two monosaccharides, use their two anomeric carbons. Carbon number 1 of glucose is called the anomeric carbon. For fructose, the carbon number 2 is also anomeric carbon. Now, in the previous examples, just like in lactose, remember, lactose is made up of galactose and glucose. In lactose, only the galactose uses its anomeric carbon. In glucose, the anomeric carbon is not part of the linkage. So the anomeric carbon of glucose here is considered to be free because it does not participate. So I repeat, in lactose, galactose uses its anomeric carbon in forming the beta 1,4 glycosidic bond. However, the glucose, its anomeric carbon is free. Now going back to sucrose, here the anomeric carbon of glucose is used in forming a bond. So this is the anomeric carbon, the first carbon. And the anomeric carbon is used to form a bond with fructose. As I said, the anomeric carbon of fructose this time is carbon number 2. And the anomeric carbon is used to form a bond. That's why sucrose is generally called non-reducing sugar, because sucrose does not have free anomeric carbon. Unlike lactose, lactose has a free anomeric carbon. Where is the free anomeric carbon? In the glucose molecule. That's why lactose is considered as reducing sugar, whereas sucrose is non-reducing sugar due to the absence of free anomeric carbon. Now let's go to polysaccharides. Polysaccharides are molecules that have many monosaccharides bonded together by glycosidic linkages. They are also known as glycans. Polysaccharides may be homoglycan or homopolysaccharide. That means they have only one type of monosaccharide present. And another polysaccharide is called hetero. That means more than one type of monosaccharide is present. Let's see this polysaccharide. Now, let's categorize them according to the following. We have what we call storage polysaccharide. When we say storage polysaccharide, they are used as energy source in the cell. Okay? Storage of energy. The first in the list is starch. Starch is produced by plants via photosynthetic processes. So starch is a polymer, as I said earlier. Starch is a molecule. It's a big molecule, large molecule that is made up of many glucose molecules. The glucose molecules are bonded in the glucose molecules are bonded using its alpha 1,4 glycosidic bonds. Okay, and starch is composed of either amylose or amylopectin. The difference between the two is this. When we say amylose, it's unbranched type of polysaccharide, unbranched. Okay, wherein the glycosidic bond is only alpha 1,4. Right here, we have a representation of an amylose. Okay, we can say that this is something like a molecule that is straight or simply unbranched, no branching somewhere. So alpha 1,4, as I said earlier, when we say alpha 1, the bond in carbon number 1 is downward. So you can expect that in amylose. Another type is called amylopectin. This is highly branched. Okay, this is highly branched, wherein the branching happens in alpha. The branch happens to be alpha 1,6. It's highly branched, made up of alpha 1,4, and this should be alpha 1,6. Okay, so right here, the bond between this glucose and this glucose is alpha 1,4, but to form a bond, it needs alpha 1,6. So remember, this is the carbon number. This is carbon number 6, and this is carbon number 1. The bond between carbon 1 and carbon 6 is alpha. Okay, carbon number 1 is anomeric, remember, but carbon number 6 is not an anomeric. Okay, so carbon number 1 is pointing downward, so it's called alpha. Alright, amylose can be digested by humans because we have enzymes that can break or that can hydrolyze the alpha 1,4. We call that amylase. So in general, we can break or we can hydrolyze, we can split starch into glucose because we have enzyme called amylase to break those bonds, the alpha 1,4 and alpha 1,6 glycosidic linkages. Okay, so when we eat starch, what happens is the starch is broken down in the small intestine, and then once it is converted to glucose, the glucose will be absorbed by the cell. Another polysaccharide is glycogen. Glycogen is also a storage polymer. But this time, glycogen is also produced, stored in the muscle and in the liver. So we can store glycogen. So after meal, the glucose that is not used will be temporarily stored as a glycogen. Okay, because not all glucose will be converted into energy at once. The rest of the dietary glucose will be converted first into glycogen, so that when the glucose level is depleted, what happens is the glycogen will be converted back to glucose. That's why I said this is a storage form of energy. This is highly branched. Okay, the structure is more complex than amylose. So this is present in the muscle and in the liver. The metabolic pathway that produces glycogen is called glycogenesis. As I said, glycogen is a highly branched molecule, and the glucose units are linked by alpha 1,4 and alpha 1,4 or alpha 1,6 linkages. So highly branched, more than amylopectin. The reason why glycogen is highly branched, because when we need to replenish our glucose or blood glucose level, there will be many sites for the enzymes. So for example, here, these ends of each of the branch will be the site of the enzyme. Okay, so in order for us to replenish our glucose level very quickly, there must be many sites for the enzyme to act upon, and those ends will become the site of enzyme action. Compared to a linear molecule, let's say this is a linear molecule, okay, although you have a very long molecule, but there's only one end. Okay, this will be the site where the enzyme will act. Okay, so there's only one enzyme that will act because there's only one end. Unlike in the glycogen, there will be many sites of the enzyme. That's how glycogen is arranged. Another polysaccharide is cellulose, and cellulose is classified or categorized as structural polysaccharide. It says here they serve as a structural element in plant cell and animal skeletons. So structural polysaccharides serve that function. The first example of structural polysaccharide is the cellulose. Cellulose is composed of glucose units, okay, but this time the glucose units are linked by beta 1,4. Remember, as I said before, Cellubiose is an intermediate product of cellulose degradation. Okay? So the glucose units are linked by beta 1,4. This is unbranched homopolysaccharide. When we say homopolysaccharide, the basic unit is uniform all throughout. So it's a component of plant cell wall, most abundant naturally occurring polysaccharide. And we cannot we cannot hydrolyze the beta 1,4 glycosidic bond because we do not have, humans, we don't have an enzyme cellulase. Okay, so it cannot be broken by human for nutrition because of the enzyme cellulase. So some animals like the cow, the grazing animals, cow, carabao, horse, you name it, they have enzyme. They have enzyme that or cellulase that can break or that can hydrolyze the beta 1,4 glycosidic linkages. Alright, so they only serve as dietary fiber which can bind to lipids and carry them out of the body. So that's the role of the fiber or the cellulose. Chitin or chitin, this is a structural polysaccharide also that gives rigidity to the exoskeletons of crabs, lobsters, shrimps, insects, and other arthropods. The material or the sugar units here, the carbohydrates unit here, is what we call N-acetyl-glucosamine as monomeric. So this is the monomeric unit. This is a glucose, but it's a derivative of glucose because on carbon number 2 there is an acetyl. Okay, this 2 carbon atom here is what we call an acetyl group, and there's an amine on carbon number 2. So the acetyl is bonded to an amine. That's why it's called N-acetyl-glucosamine. And each of the N-acetyl-glucosamine is linked to one another by beta 1,4. So again, when you say beta, the carbon number 1 here is written or shown upward. Okay? Connected to carbon number 4. Alright, so this is the structure of chitin or chitin. Then we have what we call acidic polysaccharide. Some of the examples, the example includes hyaluronic acid. Hyaluronic acid contains alternating residue of N-acetyl beta D-glucosamine and D-glucuronic acid. So alternating residue. So where is the N-acetyl beta glucosamine? So as I said earlier, this is the acetyl. On carbon number 2 there's an amine group. Okay, and then N-acetyl beta D-glucosamine. So this thing here is your N-acetyl beta D-glucosamine, and it's connected to it says here D-glucuronic acid. So this is the glucuronic acid. Remember in the reaction of sugar, if the carbon number 6 is oxidized to carboxylic acid. Remember, this is the carboxylic acid. The glucose becomes glucuronic acid. So this is the glucuronic acid. Glucuronic acid. So beta, okay, beta, this N-acetylglucosamine is connected to glucuronic acid via beta 1,4. Okay, and its link again, this glucuronic acid is linked again to N-acetylglucosamine. So it's alternating residue. Beta N, this is beta N, N-acetyl, beta glucosamine, this molecule is connected to glucuronic acid. The glucuronic acid is linked again to N-acetyl, glucosamine, and so on. So that is hyaluronic acid. It's a molecule that serves as lubricant in the fluid of joints, and they are also associated with the jelly-like consistency of the glassy humor of the eye. Next is a heparin. This is also an acidic polysaccharide. This serves as anticoagulant. It helps to prevent blood clots. It binds strongly to a protein involved in terminating the process of blood clotting, thus inhibiting blood clotting. So it's composed of repeating units of D-glucuronate 2-sulfate and N-sulfo-D-glucosamine 6-sulfate. So where are those molecules? D-glucuronate. So this is the D-glucuronate. Remember, this is an acidic because the carbon number 6 here becomes carboxylic acid, COOH, or this is carboxylic acid. Okay, that's why it's glucuronate. And on carbon number 2, there's a sulfate. So that's why it's called D-glucuronate 2-sulfate. And this molecule here is linked to another carbohydrate derivative known as N-sulfo-glucosamine 6-sulfate. Okay, on carbon number 2, there's an amino group, and connected to that amino group is sulfo or sulfate or sulfite group. And then on carbon number 6 also, there's another sulfo group. Okay, so that's the structure of, or I should say, repeating units of heparin. But of course, you have to think of it that this is a polysaccharide. That's why there's a square bracket, then N, meaning that is a polysaccharide, okay, or a polymer. We have also some carbohydrates derivatives, and when you say carbohydrate derivatives, carbohydrates with some other organic molecules. Example is glycolipids. Glycolipids are carbohydrates with lipids bonded to it. So glycolipid, the lipid molecule that has one or more carbohydrates or carbohydrate derivative that is covalently bonded to it. So these glycolipids, their function is to facilitate cellular recognition. So one of the examples of glycolipid is a glycocerebroside. Okay, here in this structure, you have here a glucose, that's the carbohydrates, and then there's a this portion here is lipids. It only shows R group but this R group is made up of long carbon chain. Okay, that's why it's called a lipid. And right here, this portion here is the sphingosine group. We will see that sphingosine in the coming lecture on lipids. So the whole thing is what we call a glucocerebroside. Then we have glycoproteins. These glycoproteins, they are actually abundant on the extracellular space on the surface of the cell. Protein molecule that has one or more carbohydrates or carbohydrate derivative that are covalently bonded to it. So they are called glycoprotein. The lipid or the protein part of the glycolipid or glycoprotein is incorporated into the cell membrane, and the carbohydrate part as a marker on the outer cell membrane surface. So they are involved in the cell-to-cell recognition or cell-to-molecule recognition. Cell recognition generally involves the interaction between the carbohydrate marker of one cell and the protein embedded into another cell membrane of the other cell. So these glycoproteins serve as marker, or in order for the cell to recognize the molecule, and in some cases for the cell to recognize the other cell. Okay, so the glycoproteins can be an O-link glycoprotein or N-link glycoprotein. Okay, O-link means the particular protein or amino acid is linked to an oxygen, but an N-link glycoprotein, the protein is linked using its nitrogen atom. O-link, the protein uses its oxygen atom, but an N-link, the protein uses its nitrogen atom.